Thank you very much. Thank you for the organizers for having me, and thank you, everyone, for coming to hear this talk about work we published in March of 2011, and some more recent extensions I'll talk about as well. So we're interested in the question of understanding the human genome sequence and how the static sequence can encode very different types of cells within the same organism, but all sharing the same static DNA sequence. And the focus of our work is epigenomics and chromatin, specifically the modifications to the histone tails around which the DNA is wrapped around these histone proteins, forming the DNA protein complex called chromatin. And now with ChIP-seq, we're able to map genome-wide the location of these modifications for dozens of different modifications across a number of cell types. And this raises computational questions of how best to model and analyze this type of data. And what we were interested in doing was taking multiple histone modifications and giving an annotation to the genome based on this type of data. And we were interested in learning in an unsupervised way based on the combinatorial patterns. And this work we first did apply to a single cell type. And our model was based on a multivariate hidden Markov model, where we assume that there's underlying unobserved genomic elements. And then at each location in the genome, we observe a certain set of histone modifications. And we binarize the data to call a presence or absence of the histone modification data. And then we modeled the mission distribution of the HMM as a product of independent Bernoulli random variables, which gives the probability of observing some specific combination of histone modifications at some location in the genome. And using the HMM also gives you spatial information. So this was previous work to the paper I'm going to be talking about. And we applied this to data in T cells to about 40 histone modifications. And what this figure is showing you is, over here, each row corresponds to the state of the model, each column one of the inputs, and the intensity, red means the higher likelihood of that modification being present when you're in that specific state. So then you can go across the genome and annotate locations to the specific states, and then determine enrichments for certain genomic categories, and then come up with candidate annotations for each location in the genome. So this was applied in this one specific cell type, CD4 T cells. But as I was saying in our motivation, we're interested in gaining some more insights into how the static sequence can code for very different types of cells. So as part of the ENCODE project, Brad Bernstein's ENCODE production group mapped a set of 8 histone modifications and insulator protein CDCF at the time across 9 human cell types, designed to be a very diverse set of cell types. So then what the raw data looks like at some location in the genome is you have different cell types and then different signal tracks for each of these cell types. And we needed some way to simplify this type of data. And we used the similar modeling approach as you saw before for the single cell, but what we conceptually did was we concatenated each of the cell types to make a genome which is essentially 9 times longer. And what this allowed us to do was have a common set of state definitions across each cell type, but that the state assignments across the cell types could be dynamic. And what you're seeing here are the rows corresponds to the state of the models, again, in the columns of marks, and the numbers here indicate the probability of observing that mark when you're in that state. And then these are enrichments for various categories, and then the candidate annotations we've given. For example, the red is going to correspond to active promoters, the orange will be candidate enhancer elements, strong enhancers, and blue insulators, green transcribed, and gray K27 polycomb-associated repressive regions. So this is an example of what an annotation at one genomic location across all the cell types could look like. So what you're seeing here are at the top 4 cell types. And each row is showing the mark intensity for one of the inputs. And we can summarize that data with a single state or color at each location. And then you can, because the states are defined consistently across the cell types, you can quickly scan some location in the genome and see the state assignments associated with this. We were interested in understanding at a more mechanistic regulatory level what could be associated with these cell type specific differences, where at some location you might have an enhancer in one cell type, but at some other location you're not observing that enhancer being present. So this is giving an example here where it's showing each row corresponds to a cluster of locations in the genome, in this case where the H1 human embryonic stem cell, you have an enhancer, but you don't see it in other cell types. In this cluster here are locations where you tend to only have an enhancer K562, and in this cluster it's locations which have enhancers in both K562 and GM12878. And what we're showing here is that of locations in the genome assigned to this cluster, they tend to be enriched for instances of known motifs of the transcription factor OCT4. So we started with the library of PWMs and scanned the genome for instances of that transcription factor's motif. And then that's indicated by the red for the motif enrichment. And we can also say the yellow indicates that when you see in H1 embryonic stem cells, you also have OCT4 expression specifically expressed in that cell type. So in this case, we have an agreement for the motif for OCT4 being specifically enriched in H1 embryonic stem cell enhancer clusters and it being specifically expressed. So we can predict OCT4 as an activator. Conversely here, GFI1 is also specifically expressed in these two cell types, but its motif is depleted. So here we can make the prediction from the data that GFI1 is acting as a repressor for this pair of cell types. And we more systematically did this through all the combinatorial usages of cell types and making predictions of what are the key transcription factor activators and repressors based on this framework. Another question that comes out of this data is we can make these calls about enhancers at specific locations, but then a natural question is what is its target gene? And with one cell type, you don't have any additional information just from the chromatin data of which gene it might be more likely regulating. But once you correlate the chromatin data with gene expression data, you can start having additional statistical evidence. For example, if this location in the genome had a K27 acetylation, which is a mark of active enhancer, specifically in the two cell types where this gene tends to be higher expressed, and in this case it's low, not as correlated, then this gives us some statistical evidence that this enhancer is more likely regulating the gene at the left. And we had a computational method which would integrate multiple enhancer modifications, the correlations, and information about the distance and the specific chromatin state assignment using a logistic regression classification framework to give probabilistic linkages scores. So another thing we did with this data, for which there's a lot of interest now, is that genome-wide association studies are implicating certain locations in the genome as being functionally associated with disease without any additional evidence that these locations in the genome are functional. They're occurring away from any known genomic annotations. But now that we had annotations of the genome based on the chromatin states in 9 specific cell types, we systematically intersected the very top hits curated into the NHGRI GWAS catalog with our chromatin states. And we focused here specifically on the states we associated with being strong enhancers. And after doing all the corrections for multiple testing, we found that our top 10 ranked associations of studies and cell types were significant, and they made biological sense, and that the top SNPs associated with the erythrocyte phenotypes enriched in enhancers associated with the K562 cell line, blood lipids and cholesterol associated with HepG2, which is a liver-related cell type, and immune-related disorders associated with enhancers from the GM12878 lymphoblastoid cell type. And these annotations can be used as a resource going forward to help prioritize SNPs based on, if you had multiple SNPs in linkage disequilibrium with a similar p-value, the chromatin data could give you information as to at a higher resolution, which locations are more likely functional. And you could further make predictions based on the motifs that are being created or disrupted based on what I presented earlier of predicted activators and repressors in this cell type. So the intersection with the genome-wide association studies gives some suggestive evidence of the functional importance of some of these enhancer locations. We also more directly experimentally validated some of these enhancer regions. So the figure on the left is showing one of the predicted activators, HNF4, the motif. And you can see if we aggregate the signal around one of the histone modifications, H3K4ME2, there's a dip right where the motif is, suggesting that the transcription factors tend to bind there, and then there's some nucleosome exclusion. And we tested 10 locations that we were predicting as enhancers, as well as the wild type containing this motif, and also permutations of the motif where we disrupted the motif. And these tests validated in this luciferase reporter assay that both our predicted enhancers were functional and that this motif disruption was sufficient to disrupt the activity of the motif. So this was all work we had done up until the paper. Since then, we've now done work doing high-throughput experimental testing of candidate enhancer sequences and in a framework where we can do de novo discovery of the driving motifs. So in some sense, it wasn't completely satisfying that we could make tens of thousands of enhancer predictions across the genome, but then we were only experimentally testing 10 of them. So what we're leveraging now is recent technology spearheaded by Tarjei Mikkelsen that was recently published. And the idea here is this massively parallel reporter assay, where you can have some, in this case, they took some sequence of interest, did a large number of mutations to it, barcoded the sequences, and then transfected it, and then sequenced which in front of the, so that the specific enhancers which are active, you would get the barcodes corresponding to them when you did the transfection and sequence. So this can tell you which enhancer sequences are driving expression. So in this paper, they focused on one specific sequence and did a large number of mutations. We can use the same technology to test lots of different sequences. So the specific design we have here, in this case, we took our enhancer calls, several hundred of them, and we tiled them, walking 30 base pairs from the center of the dip of the histone modification. We had 4 sequences to the left and 4 sequences to the right. And we had some set of locations which were enhancers in HepG2 and not in K562, and then another set which were enhancers in K562 and not HepG2. And you can see here when we transfected into HepG2, the enhancers on aggregate were active and that the control, much more than the control, and specifically close to the center of the dip. And this is showing another view of the data where we did 2 biological replicates. Each actual sequence was replicated in the assay 24 times. So, but it's showing that it's extremely quantitative, the readout we get. And this is giving you more than 1,000 sequences. And so that's for 2 biological replicates. Now, if we compare in average the same sequence and just doing this 30 base pair offset, you can see that it's sufficient to severely disrupt the enhancer readout. So what this now has given us is that it's very high resolution. It's isolated 30 base pair sequences that are sufficient to drive enhancer activity. And what we have is, we have a large number of sequences at a very high resolution that are sufficient to drive the reporter assay. So in this case, it makes the computational discovery of de novo motifs very easy when you have so many sequences at a very short that you know are sufficient to drive gene regulation. So we ran meme in this case, focusing here on the positive sequences, and then we also, as a negative set, took this other set which had a lower readout. So these numbers here are showing this sequence, what its reporter value was in the 2 replicates, and the green is the replicate values for the same sequence, but just this 30 base pair difference. So you can see here that these were the motifs discovered in HepG2. It found 4 significant motifs. None of these were found in the negative set. And when we matched to the best known motif in a database using TomTom, you can see there was very high statistically significant agreement that well matched. And these correspond to known important liver regulators, and this is activator protein 1. And when we did this, use the same sequences but put it into K562, you can again see a very good agreement between the motifs which were discovered and what was annotated in the database. I now want to talk about an expanded chromatin state model and intersecting it with transcription factor binding. So we were part of the ENCODE consortium, which is working on a coordinated set of cell types. So in addition to the histone modifications and CDCF, which the Bernstein production group was generating, there was also other marks of open chromatin and POL2. So we learned a common set of 25 states across these 6 cell types. And just want to mention briefly some of the promoter, enhancer, and insulated related cell types. And we've been focusing, going in depth on some analysis of now, the ENCODE consortium has mapped over 119 regulators, transcription factors, and other chromatin regulators. And can we have some way of interpreting these different transcription factors in the context of the chromatin states we've defined? So what we've done now is we've done an analysis where we've systematically gone across our chromatin states and for each regulator, and we've asked how enriched it is for each of the chromatin states and show its relative preference for each state. So what you're seeing here are a group of regulators which strongly prefer promoters, and a group over here which strongly prefer enhancers, and another group of regulators which strongly prefer insulator regions. So this is always comparing the matched cell type that the regulator was matched in to the matched chromatin state. We've also done an analysis where we've expanded it, comparing the locations of the binding of the regulator to all 6 chromatin states, even if it wasn't in a matched cell type. And what you can see is there's a group of regulators which are all highly enriched for promoter regions and insulator regions, no matter what cell type was carried out in. While there's another group of ChIP-seq experiments. So the rows here are ChIP-seq experiments. The columns are states, and then the smaller columns correspond to specific cell types within that state. You can see enrichments for the cell type enhancers here corresponding to the 6 different cell types considered. And these rows were computationally ordered in an unbiased way to maximize the similarity between the experiments. So another way this state is useful, the chromatin state is useful in interpreting transcription factor binding, is considering the co-occurrence patterns of transcription factors. So if you look at where pairs of transcription factors bind, there tends to be a lot of co-binding and a lot of structure to this data appears. But when you put it in the perspective of the chromatin states, it turns out that a lot of this transcription factor binding could potentially be explained by a shared chromatin state preference. So what we're saying here is you could say that 2 transcription factors might share a preference for active promoters or active enhancers, where A and B prefer the transcription start site and C and D prefer enhancer regions. So what we did was we conditioned on each transcription factor's individual chromatin state preference to give what's the expected pairwise overlap given you know each factor's individual preference. And once we do that, we see almost all the block structure is gone and the enrichments are much lower. We went from a 500-fold scale to here it's a maximum of 50-fold. And what this coloring here indicates is the cluster assignment from before, where the red corresponds to the more promoter preferred and the orange the more enhancer preferred clusters of the TFs. And this is showing another example. In this case, before is in HepG2. This is in K562 of the raw enrichments and after conditioning on our chromatin state assignments. And this can also be useful to highlight some exceptions, which even after conditioning still show more pairwise enrichment than you expect, such as things associated with pol 3 or certain heterochromatic repressive binding complex. So to summarize, I presented to you an approach for chromatin state modeling that could be used to annotate genome-wide elements across multiple different cell types. And I showed you some recent work where we've experimentally tested in high throughput a large number of these these enhancer state calls, showed how it can be used to discover de novo driving motifs of enhancer activity, and showed some results comparing these expanded set of chromatin states with genome-wide maps of transcription factor binding experiments. And that could be used to describe most previously observed co-binding. It's important to note it doesn't necessarily mean that all, it could be the case that some of the co-binding is causing a similar shared chromatin state preference, but it's saying that it is sufficient to explain it, the shared chromatin state preference. So I'd like to acknowledge the other people involved in this. So this was a joint effort between Brad Bernstein's ENCODE production group and Manolis Kels's lab, and Tarjay Mickelson did the massively parallel reporter assays. And the ENCODE consortium provided a lot of the data. So thank you, and I'd be happy to take questions.